Hello everyone and welcome to today's webinar titled Serological and Cellular Approaches to COVID-19 Research Using Flow Cytometry. This webinar is part of the 2021 Microbiology Virtual Week, and I am Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Luminex. For more information about Luminix, please go to www.luminexcorp.com. So let's get started. I want to begin by reminding everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during this presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of our presentation. And if you have any trouble seeing or hearing this presentation, just click on that Ask a Question box located on the far left of your screen. Now, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located on the top right-hand corner of your presentation window and follow the process to obtain those credits. I now present today's speaker, Dr. Kamala Tyagarajan, the Director of Flow Cytometry Assays and Applications. For complete biography of our speaker, please visit the Biography tab at the top of your screen. Dr. Tyagarajan, welcome. You may now begin your presentation.
Good morning, everyone. Welcome to today's presentation on serological and cellular approaches to COVID-19 research using flow cytometry. My name is Kamala Tyagarajan, and I'm Director of Flow Cytometry Assays and Applications at Luminex Corporation. During this presentation, I would like to discuss methods in COVID-19 research, especially with regards to SARS-CoV-2 serology and cellular assays enabled by flow cytometry. We will be following the agenda shown on the slide here for this presentation. We'll start out with discussing COVID-19 research needs and the role of flow cytometry in such research. We will spend the bulk of the talk on the Guava SARS-CoV-2 multi-antigen antibody assay, describing assay features and its importance in context of SARS-CoV-2 serology. We'll show several applications of using the assays for various studies, including those of applications to vaccinated samples. And then we will spend a short time on looking at how flow cytometry can enable cellular assays relevant to COVID-19 research. We will then conclude with a brief summary. In mid-2021 today, we stand still buried amidst the COVID-19 pandemic, with the SARS-CoV-2 virus continuing to persist and predominate worldwide. This has created an acute need to assess the immune response and to develop strategies to monitor, tackle, and overcome the disease. Numerous publications have shown that cellular and serological responses are crucial to understand the nature of the immune response in both natural infection and post-vaccination studies. Several of these publications have shown that flow cytometry is a powerful technology that can provide the capability to address many of these questions. Questions in COVID-19 research span a wide range. These include questions on how does the virus impact the immune system, how does the immune system respond, and how does one bolster the response with vaccine strategies and drug discovery approaches to modulate and eliminate the virus. We'll touch upon some of these areas during our presentations. Techniques such as phenotyping studies, phenotyping of T cells or lymphocyte subsets, and changes in responses in immune cell subset or PBMCs on stimulation with viral antigens have been really crucial in understanding the nature of cellular immunity. Similarly, another major area of study is serology and the development of SARS-CoV-2 specific antibody response to understand humoral immunity. Several researchers are thus interested in obtaining information on both cellular and serological aspects in their investigations. Currently, many of these studies are being performed on separate platforms, and technologies that can enable obtaining both cellular and serological information on the same platform can provide a great deal of flexibility and convenience to researchers. Flow cytometry is a technology that can allow one to perform multiparametric cellular assays as well as bead-based immunoassays using characteristics of scatter and fluorescence. Luminix provides a range of flow cytometry platforms that can fit different needs. These include smaller, compact platforms with customized assays, such as the Guava Muse cell analyzer shown in the first column. We also provide multiparametric platforms such as the Guava EasySight flow cytometer and the Amnis cell stream flow cytometer, which are equipped with multiple lasers and many detection channels. Our advanced systems include the imaging flow cytometers, such as the Amnis FlowSight and the Amnis Image Stream system, that combine the power of imaging cytometry along with flow cytometry to provide insightful information. The choice of platform can be dictated by throughput, needed number of parameters that need to be detected simultaneously, budgets, and expertise of personnel available. In the next part of the presentation, we will be mainly focusing on the platforms based on microcapillary flow cytometry, which is mainly the Guava Muse cell analyzer and the Guava EasySight flow cytometers and its applications to serological research. Microcapillary flow cytometry. Microcapillary flow cytometry platforms have great utility for infectious disease research. Microcapillary cytometry, shown on the left, differs from traditional flow cytometry on the right in that it does not utilize sheath fluid. The sample is aspirated directly from a sample tube or well through a microcapillary flow cell, and it's interrogated by a laser as it goes in single file. Data is collected from the detection channels as the sample flows through. This, when combined with the positive displacement syringe pump, allows for the determination of absolute counts, which is very useful in many cellular assays. This unique design leads to smaller and more compact size of instruments and results in them being able to place them in convenient locations such as biological hoods if needed. The technology offers many advantages. It results in the need for reduced sample sizes and fewer number of cells per analysis. And this can be very helpful when only precious amount of infected samples may be available. Next, the system generates low biohazardous waste, and this, as you can imagine, is very useful when dealing with infectious samples. But most importantly, these systems also provide absolute counts without needing to use external beads, which is highly useful in characterizing immune cell populations or normalizing cell number. I would like to now move on to discuss a new assay we have developed for flow cytometry, the Guava SARS-CoV-2 multi-antigen antibody assay and its applications to COVID-19 related questions. Serological assays have played a crucial role in COVID-19 research. These assays have shed light on the magnitude and persistence of the immune response in different studies, such as natural infection, epidemiological and surveillance studies, and in the development of treatment strategies and development of therapeutic drugs. The assays continue to be important to study vaccination response in different subpopulations and the persistence of response in different groups, with more strategies such as booster strategies coming in. As people consider different assay types to use, they should consider if the assay of choice can provide comprehensive information on the different antibodies present in the samples against different antigens, whether they need isotype-specific response information, and if the assay of choice has a wide dynamic range to span the entire kind of samples that they are studying. So what are the key antigens of interest to people studying SARS-CoV-2 antibodies? The SARS-CoV-2 genome encodes several proteins, and well known among these are the nucleocapsid protein, the spike protein, membrane glycoprotein, and the envelope protein. The nucleocapsid protein is an abundant and important structural protein of SARS-CoV-2 virus. This participates in RNA packaging and viral particle release and is highly immunogenic. Antibodies against this protein indicate exposure to SARS-CoV-2, and anti-N antibodies have been detected with high specificity in early stages of infection. The S protein plays a critical role in viral infection and pathogenesis, and it's a critical target for the induction of antibodies, particularly neutralizing antibodies. The S protein contains two subunits, the S1 and the S2 subunit. The S1 subunit contains the receptor binding domains that mediates the binding of the virus to the ACE2 receptor. People have used multiple forms of the S protein, the RBD domain, the full-length S1 plus S2, or partial S1 domain for different serological tests. Assays that are targeting multiple antigens and fishing for the different antibodies in parallel can provide a better understanding of the nature and magnitude of the antibody response. And this aspect has become particularly important as we'll discuss in dealing with vaccinated samples and also in dealing with variants. So multi-antigen serology has become key to providing more detailed information on what's going on with the immune response. Multiple antibody isotypes have been shown to be produced in response to the SARS-CoV-2 virus in plasma and serum samples. Several papers have shown the presence of IgM and IgA isotypes in addition to the IgG isotypes, and also establish that these additional isotypes contribute to the immune response mechanism and neutralizing activities. It is important to note that different isotypes may predominate depending on the time that has passed post-symptom onset, and this can be seen from the chart on the left, where we can see that the IgM and IgA isotypes tend to rise early and then fade away a bit faster, while the IgG isotype rises up a little slower but has a more sustaining response when we look at samples at different stages post-symptom onset. Thus, tracking the dynamic changes for the isotype-specific response may be important to understanding the full spectrum of response. It's also interesting to note that while there are many assays available today for IgG detection against SARS-CoV-2 antigens, fewer assays are available for IgM and IgA isotypes, and often studies are pooling data sets from different platforms or different assays to get the information together, which is not ideal. I'd like to next discuss a new assay for SARS-CoV-2 antibody detection called the Guava SARS-CoV-2 multi-antigen antibody assay. The assay is a bead-based immunoassay and uses a simple sample prep protocol followed by acquisition by flow cytometry. The assay allows for antibody detection against multiple SARS-CoV-2 antigens in parallel and permits the detection of IgG, IgM, and IgA antibodies using the same kit. Flow cytometry-based acquisition and analysis can be performed either using the assay on simplified platforms like the Guava Muse Cell Analyzer or on the Guava EasyCyte HT platforms. It can also be used on a third-party instrument equipped with a blue 480 nanometer or green 532 nanometer laser. Outputs from the assay can be used to interpret the presence of antibodies in the sample being studied. The assay kit includes a microsphere mix, buffers, and PE conjugated detectors against the three isotypes. The multiplex bead assay kit includes negative beads and 3 beads against the 3 SARS-CoV-2 antigens. And the antigens include the N bead, which is conjugated to the nucleocapsid protein, the RBD bead, which is against the receptor binding domain of the spike protein, and the S1 bead, which is conjugated to the S1 subunit of the spike protein. The 3 isotype-specific detectors are conjugated to PE. The protocol is really simple. Beads are incubated with diluted sample, and this is incubated for 30 minutes. There's a simple wash step where the supernatant is removed, and then detectors are added to the sample, followed by 30-minute incubation, removal of supernatant, and then the sample is ready to read on the instruments. The sample can be prepped in tubes or plate, depending on the platform of interest. The reagent kit can be used with multiple cytometers. On the Muse Cell Analyzer platform, there's a SARS-CoV-2 antibody assay specific software module, and this module guides the user through specific steps of data acquisition and interpretations. Data outputs include plots and then median fluorescent intensity information. On the Guava EasySight 12HT cytometer, Insight software and a settings file can be utilized to obtain similar results needed for interpretation. On third-party cytometers, one can use the typical acquisition software and set it up for data acquisition and analysis. Thus, there's a visual output of results as well as median fluorescence intensity data provided for data interpretation. The slide here shows you examples of data obtained by analyzing serum samples on the left and plasma samples on the right using the Guava SARS-CoV-2 multi-antigen antibody assay. For negative samples, we observe from the plots that all 4 beads line up and there is no shift of the beads in the analyte response direction. This means there are no antibodies against SARS-CoV-2 antigens present in the sample. For positive samples, we can notice that there's a shift of 1 to 3 beads in the analyte response directions, indicating that antibodies against this particular antigen are present in the sample. By using the isotype-specific detectors, one is able to obtain information on the presence of IgG, IgM, or IgA-specific isotype in that particular sample. We next looked at antibody response between negative and positive samples for a larger data set using the assay. These included serum samples from 28 negative donors who were mainly pre-September 2019 collected samples. The data set also included samples from 27 donors which were confirmed positive based on SARS-CoV-2 molecular or PCR test results previously. Samples were prepped in tubes for analysis in the Muse Cell Analyzer and the third-party cytometer, and they were prepped in plates for analysis on the Guava EasySight 12HT system. As you can see in the plots, median intensity information is plotted for each of the bead populations. For negative samples, we notice that there's little or no response of the beads, minimal intensity being observed for each of these beads. And this data is shown only for the Muse Cell Analyzer, but similar results were obtained on the other system. For the positive samples, what we notice is that there is distinct intensity observed for 1 to 3 antigens for the different donors that were studied. And this is seen across the 3 systems. So data here shows us very strongly that we can cleanly identify positive from negative donors, and also the same nature of detectability can be obtained using the kit across the different platforms. One can see the IgG response also more visually. Here, response from serum samples from different donors is shown. The days on top of each sample indicate when sample is drawn post first symptom. The data shows that there is response in samples even starting at 6 days and all the way up to 133 days in the sample studied. Thus, in each case, we can see that there is a clear and distinct shift of populations being observed and identification of the samples as being positive by the assay. We next looked at antibody response in samples collected from different positive donors at different periods of time post-symptom onset. 2 sample types were studied. One set included 14 donors, which were collected within 14 days of first symptom. The second set included about 20 donors, and these were positive and collected at greater than 14 days post-symptom onset. As one can see in this plot of the intensity for these bead sets, we notice that in almost all samples show good response for antibodies against multiple antigens with this assay. Interestingly, response can be seen in several donors less than 14 days, allowing for antibody detection. And all donors greater than 14 days show response from 2 or more antigens. The high sensitivity from flow cytometry might thus allow for earlier and clear-cut identification of the presence of these antibodies. We also looked at the presence of antibody isotypes in a larger sample pool. Here we show results from detection of IgG, IgM, and IgA isotypes in positive samples that were positive by molecular test results, and comparing that response with pre-September 2019 negative samples. While it's not surprising to see IgG reactivity as shown in the first plot for a large number of samples, it's interesting to note that there is a substantial number of samples which show IgM and IgA isotypes against the different antigens in particular samples. Thus, the study of isotype presence is very significant in understanding the complete nature of the immune response, so we don't miss out understanding the significance of the effects. One can see these effects more visually for antibody isotype detection in a range of samples from 43 to 260 days. And what one notices is that while we see IgG antibodies against the different antigens over the full span of samples that we are observing, IgM and IgA antibodies are observed in fewer samples, and this declines as the sample ages. Let's take a look at why looking at antibody isotypes may be important in different studies. Here we show an example of donors at multiple duration post first symptom. Donor 1, which is 5 days post symptom onset, shows the presence of IgG antibodies against the nucleocapsid antigen. But what is really striking is that this donor shows very high levels of an IgM antibodies against the RBD antigen. It also shows the presence of an IgA antibody against the nucleocapsid protein. And it's interesting to note that this particular sample showed very high neutralizing activity in an in vitro viral neutralization assay. Donor 2 is another kind of sample where the donor is 13 days post-symptom onset, and samples from this donor were analyzed with the assay, and what we notice is that there appears to be good levels of IgG, IgM, and IgA isotypes all present in this particular sample. If we look at another donor, donor 3, which is 124 days post-symptom onset, we notice that this sample is predominantly all IgG, and there's negligible amounts of IgM and IgA antibodies present against any of the antigens. And this is in line with our understanding of the decline of IgM and IgA isotypes as these samples age. Thus, depending on age of sample and status, different antibody isotypes could predominate and may be very important in drawing conclusions about sample sets. One can also use the assay to study the development of the isotype response over different days for the same donor, that is, application to longitudinal studies. Over here, we have the same donor who is positive by PCR for SARS-CoV-2 infection, and sample was collected over multiple days from 9 to 59 days from the donor. The samples were then assayed using the Guava SARS-CoV-2 multi-antigen antibody assay and run on the EasySite 12 HT system. The heat map features of the Insight software gives you a very nice visual display about the different levels of each of the antibodies against the 3 antigens for each of the different isotypes. And this allows you to get a quick visual feel for how the isotype levels are. One can also look at this data graphically, and for simplicity, the data for the N antigen is shown in the plots on the right. As you can see in these plots, you can see that the IgG response shows a more classic profile and is sustaining. The IgM and IgA beads show response and presence of these antibodies in earlier time frames, which then tend to taper off as time progresses. Data such as this can be conveniently followed by the kit and analyzed by flow cytometry. I would like to now move on to the applications of the assay to vaccinated samples. As the world intensifies its efforts on vaccination strategies, serological assays continue to play an increasingly important role. Anti-SARS-CoV-2 antibody assays can aid not only the development of the vaccine itself, but also in determining the long-lasting impact. The vaccine strategies today include DNA or mRNA coding for the SARS-CoV-2 spike protein. They also include protein or peptide-based approaches from the SARS-CoV-2 proteins, or they include engineered viruses, that is, viral vectors expressing, for example, the spike protein. There are also classic approaches like using inactivated or weakened SARS-CoV-2 virus. So as people choose the best assay to study samples from vaccinated donors, they need to look at the vaccine type and the antigens to which they would expect to see an antibody response. For example, some of the mRNA-based vaccination strategies are against the spike protein or the receptor binding domain of the spike protein. And this will give rise to antibodies against these proteins, but these tests will be missed by an antibody test that is based on the nucleocapsid protein from the SARS-CoV-2 virus. For strategies such as inactivated or weakened virus, one would expect to see responses against the nucleocapsid protein, spike, and RBD protein. So one really needs to understand what vaccine was given to a donor and what is the expected response. One also needs to make sure that the assay of choice has a dynamic range which can detect samples from early infection to high level of antibodies which can be observed in some vaccinated donors. So what are some of the questions of interest in the study of vaccinated samples? Several studies have used serological assays to look at sample type being researched and confirm the immune response and presence of expected antibodies on vaccination. The capability to distinguish between vaccinated samples versus naturally infected samples has become very important in understanding and monitoring breakthrough infections. Another crucial area of study is elucidating immune response differences in different immunocompromised subgroups. This includes individuals with cancer, transplant recipients, people with autoimmune diseases, and people on immunosuppressant drugs. In many of these studies, elucidating the duration, persistence, and response is very important to understand general trends. These studies are also very important in illuminating requirements or needs for future vaccination or booster shots. Here we show examples of applications of the assay to serum samples from vaccinated donors. Donors include people who have had either Pfizer or Moderna vaccines as indicated. The upper plot shows the IgG response. The middle set of plots represents the IgM response, and the third set of plots represents the IgA response in these samples. The plots immediately show the presence of IgG antibodies against RBD and S1 antigens across the different donors, which is what we would expect to see. Even visually, one can note that there's varying levels of antibodies one can observe between the different donors. The donors do not show any presence of IgM antibodies. but several donors showed the additional presence of IgA antibodies against the RBD and S1 antigens. We next studied the antibody response in samples that we obtained from J&J vaccinated donors. In the few samples we studied, we noticed that while a couple of the donors had the expected response against the S1 and RBD protein, as would be expected with this kind of vaccine, one of the donors had no IgG, IgM, or IgA levels. And it was interesting to note that this non-responsive donor had an autoimmune condition. The multiplex multi-antigen antibody assay can be very helpful in identifying donors that might have had previous infection. In the example plots here for the Pfizer vaccinated and Moderna vaccinated donors, in addition to the RBD and the S1 bead response we would have expected to see, we are also seeing a response of the N bead, indicating antibodies against the nucleocapsid protein are present in these samples. And this is indicative of the fact that these donors might have had natural infection, due to which we are seeing antibodies against the nucleocapsid antigen. These kind of multiplex antibody assays can thus help distinguish between a vaccinated, uninfected sample and samples with suspected natural infections for certain vaccination types that are against the spike or RBD proteins. The figure here shows the median fluorescence intensity response using the Guava SARS-CoV-2 multi-antigen antibody assay on non-vaccinated and vaccinated donors. The first set of plots here is from infected donors which are non-vaccinated. And what we notice here is that we see good response from the N bead, the RBD, and the S1 bead for most of these donors. If we look at the Pfizer vaccinated samples, we see there is strong RBD and S1 response in most donors, but a donor or two has N response in the sample tested, which indicates likely prior infection. We can also see a donor with low intensity of signal for the RBD or S1 response. On the Moderna samples too, we see a strong RBD and S1 response, and the presence of at least one donor who appears to be previously infected. Thus, we see a spread of IgG responses from donors, not all donors showing high intensity. One can also study trends in antibody development before and after first and second shot of vaccination. In this study here, we evaluated a few donors where we had samples from before vaccination, after the first vaccine, and after the second vaccine. As we see in the plots from these donors, there's different trends in IgG response on vaccination after the first and second doses. But we do see that there's a boost which is achieved after the second vaccine. It also seems like the levels of IgG produced between the different donors are also very different. This kind of data can be compared in the plots as shown in this figure, where the plots show the response from each donor for antibodies against each particular antigen in the vaccination studies. We notice in each case that the RBD response is dominant and is boosted significantly with the second shot. Studies such as this in larger donor pools might provide a more complete picture of the trends on what's going on with regard to the immune response in these kind of different sample types. I would like to next take a few minutes and switch to discussing how flow cytometry can provide answers relevant to COVID-19 research for cellular questions. As we discussed earlier, COVID-19 research can often require quick screening of blood and PBMC samples to obtain characterization and enumeration of subpopulations. Flow cytometry platforms that can provide absolute count information economically are very useful for the ID and enumeration of whole blood or PBMC samples. Thus, assays such as the count and viability assay or immunology assays on microcapillary platform can provide answers using very low amount of samples using assay-specific templates. The count and viability assay uses a pre-formulated mix of membrane-penetrating and non-penetrating dyes, and this allows for the sensitive detection of nucleated cells, dead cells, and debris. And thus, in the sample, when mixed with the cellular sample, it can provide distinction of viable cells from debris and distinction of dead cells from viable cells using very small quantities of sample. Similarly, the Muse Immunology assays are mix and read assays that can provide information on different immune cell populations in whole blood or PBMC samples very easily and quickly. Muse count and viability assay is a very useful assay for determining cellular concentrations and viability status in a cellular sample. This has been applied to the study of numerous cell types, including fresh and frozen PBMC samples. The assay has a dedicated software module that outputs summarized data in an easy-to-interpret format, as shown in this figure. The cell concentration outputs from the assay have been compared to predicate methods for fresh PBMCs, frozen PBMCs at multiple concentrations. And as you can see on this plot here, that you're seeing very nice correlations between the different methods. Precision of the assay has also been studied, and not only for PBMCs, but also for other cell lines. And typically what we observed is that you can obtain a percent CV of less than 10% for cell concentration and viability results. And thus this assay can be of great utility in looking at viability or concentrations of cellular samples that might be important, especially if people are preparing the samples for immune response studies or pre-screening the samples and deciding whether it's good to go to the next experiment. Absolute counts and percentages of T and B cells has shown to be very important in the phenotyping of COVID-19 samples, and CD4 and CD8 T cells in particular have been shown to undergo significant reduction with the severity of the disease. Capturing this information for all samples can be very important, and Muse Immunology assays can provide absolute counts of CD4 and CD8 T cells, total lymphocyte counts, and percentage of population using only 10 microliter of blood. Example of CD4 T cell and CD8 T cell is shown in this figure over here. Dot plot show the results for the CD4 T cell, for example, here, where we can see the upper two populations represent the total lymphocytes, and the upper right population represents the CD4 T cell population. Similarly, for CD8, the upper right represents the CD8 T cell, and the total upper population represents total lymphocyte. And this is utilized to obtain information on absolute count of the T cell, total lymphocyte count, and percentage as a percent of total lymphocytes. On the right here, we show comparison of the CD4 T cell absolute counts from Muse using microcapillary cytometry against higher-end flow platforms. And as we look at this figure, we see that across a wide range of measurements, we are seeing really nice correspondence of results. We've also looked at precision on these assays. Here is results from looking at 3 operators on 3 instruments running 4 samples and triplicates. And as we look at the percent CVs in these measurements, we see typically that we obtain a percent CV of less than 10% for a lot of these studies. Thus, the simple no-wash protocol and easy-to-use instrument with microcapillary cytometry can really enable one to quickly obtain enumeration information on T and B cell populations. One can also obtain more detailed studies and obtain enumeration information on multiple subpopulations in parallel. Here we show multiparametric analysis for TB and K populations using flow cytometry. Example is shown for CD-Chex Plus on the top and CD-Chex Low controls, stained with combinations of CD45, CD3, CD4, CD8, CD19, and CD56 and CD16 antibodies. And most of these antibodies are each conjugated to a different fluorophore and so can be looked at in different channels. The data can be easily mined to obtain count and percentage of each of these populations, and they can be then compared with other methods or other data that you've obtained. Here, for example, we have compared the results obtained to those provided by the vendor, as shown in the plots on the right. As you can see in the graphs here, the results from the EasyCyte 12HT are in blue, and the vendor results and the limits are in different colors. And as you can see from the data, the absolute count information for the various subpopulation in both sample types is well within limits provided by the vendor. Data such as this, which can be obtained in 10 microliters of blood, it can be very useful, and especially because you can perform these assays in a no-wash format in either tubes or plates. One can take this further to obtain more detailed information on naive and memory T-cell populations. Here, fresh whole blood was stained with different combinations of antibodies in a no-wash format and analyzed by microcapillary flow cytometry. In addition to being able to dissect T cells, one can see the B cells and K cells, and further one can subdivide the CD4 and CD8 T cells into naive and memory subpopulations using additional antibodies in the mix. This information can also be obtained with 10 microliters of blood, and these kind of phenotyping studies can thus be very useful in studying different donors in much greater detail. To summarize my presentation today, we discussed that COVID-19 research requires the evaluation of a wide range of cellular and serological parameters, and these methods can be enabled by flow cytometry. We also discussed that serological studies continue to be important for understanding COVID-19 related immune response, its sustenance, and the post-vaccination response. We showed you information on the Guava SARS-CoV-2 multi-antigen antibody assay and described that it's a new multi-antigen bead-based assay for flow cytometry that can provide a complete picture of antibodies against multiple SARS-CoV-2 antigens. The targets include nucleocapsid protein, RBD, and spike S1 protein. IgG, IgM, and IgA antibodies against all these proteins can be detected using the same kit. And the assay is a universal assay which can be utilized on platforms like the Guava Muse Cell Analyzer, the Guava EasyCyte system, as well as other flow cytometers. We hope that with this increased capability, the combined information from cellular and antibody characteristics on the same flow cytometry platforms can enhance and empower COVID-19 research. And finally, I would like to acknowledge all team members and colleagues who contributed to this work. The bulk of the data presented was generated by Kim Van Tran and Julie Clore. My thanks to my R&D and marketing team members who contributed to multiple aspects of this project. And finally, my thanks to the audience for their attention. Thank you.
Thank you, Kamala, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. Let's get started. All right, our first question.
Kamala, is the information on isotopes important when studying vaccinated samples, and what role do these isotopes play?
Thank you for the question, and thank you everybody for attending today's webinar. So when you're looking at SARS-CoV-2 infection, natural infection, numerous studies have shown that the different isotypes, IgG, IgM, and IgA, are observed in serum and plasma samples, and they play a very important role in neutralizing activity, for example. In vaccinated samples too, studies are showing, in particular, the presence of IgA antibodies too. Several studies have demonstrated that. And in fact, some recent studies have also shown the presence of IgA, not only in serum, but also in milk secretion. They have shown that in mucosal pathways. So the isotypes are present. And because they contribute to the immunity in the natural infection, it's expected that these antibody isotypes also contribute to the neutralizing activity that we see even from the vaccinated samples. So this has become a very important area of study where information is still being gathered because vaccination and then looking at the samples in detail is still newer, and the isotype and how do they really build up and contribute to that resistance and the immune response is still to be understood. Thank you.
Thank you. Next question we have here. What are the advantages in flow cytometry over other methods for SARS-CoV-2 serology?
That's a very good question. There are numerous methods for serology, and each of them has their own advantage. But flow cytometry provides certain unique advantages. For the biggest advantage you can get by doing the assay by serology assay by flow cytometry, for example, like we have shown, is that you can take advantage of the multiplexing that you can get with flow cytometry. You're able to look at multiple response to multiple antigens in parallel. So there's a lot more information that you are getting per experiment or per per assay that is run on the cytometer. Secondly, you have fluorescence detection. And many other methods for serology use colorimetric methods, for example. And these have some tend to have lower sensitivity, but with flow cytometry, you get the advantage of the high sensitivity that can come from fluorescent detection. And that results in actually the advantages of being able to detect samples early on. And it also comes with a broad dynamic range that is really important to look at natural infection as well as vaccinated samples. So the third kind of advantage you have in how we have the assay structured, especially by flow cytometry, is that many other methods require you to run whole plates of samples. So you need to, depending, if you had fewer samples, you may have to sacrifice certain your spots in your plate. But in a flow cytometry, if you had a fewer samples, you could run this in tubes or you could run it in plates. It just depends on what your throughput may be or how it varies from day to day. So certainly very distinct advantages with flow. But also another crucial example is that a lot of people who are doing these kind of assays are also interested in cellular immunity in addition to humoral immunity. And a lot of those assays get run on flow cytometers. And now by having this capability to run COVID-19 serology and flow cytometry, they're able to do both their humoral immunity studies as well as cellular immunity related studies on the same platform.
Thank you for that informative answer. Our next question we have here, why is serology still so important in a post-vaccine world?
That's a very good question. And in fact, the importance of serology keeps getting more and more important. With vaccination, what people are looking for is what's the immune response? Do we have the antibodies present in it, many of which studies have established can fight off infection or severe infection, right? So with vaccines, people are interested in understanding, is there that immune response? Then what is the duration? How long do these antibodies stay on? Do they go down? Do they need to get booster shots? So all of these kind of answers are aided by serological assays. The other kind of information that you can get is with relation to particular subgroups. Now, numerous studies in the past several months have established that certain subgroups of people who maybe, who have had maybe kidney transplant, cancer, or be on immunosuppressive drugs, many of these people have had reduced or sometimes no level of antibodies. And this is, again, extremely important to understand what is their immune reaction, especially with regard to antibody production is on vaccination, and what kind of strategies do we need to develop so that we can bolster that response for all people. And there may be particular treatments needed in certain cases for these kind of subpopulations, so what strategies will work better for them. So there are a lot of open questions still, and I think serology plays a very crucial role in trying to gather information and answer some of these questions, which will help us with tackling the COVID-19 and the virus itself.
Thank you. It looks like we have time for one more question. What is the utility of screening against multiple antigens approach for SARS-CoV-2 serology?
Well, that's a good question. As we saw in the talk, we had examples of a multi-antigen approach. And if you look at it, what it really tells us is it gives us information on the full spectrum or on a more comprehensive spectrum of antibodies that are present in a sample that you study. But in addition, there's also these scenarios where, for example, if there was someone who has not had the COVID-19, but they had a vaccination with, especially with any vaccine strategies that related to the spike protein, then we can see in those cases that we only see antibodies related to the spike or the receptor binding domain protein coming up. So now, if these people had natural infection or were having natural infection at the time samples were taken from them, you would see also the nucleocapsid protein response, antibodies against the nucleocapsid protein come up. So the having multiple antigens, this allows you to learn a little bit more about the full spectrum of antibodies, as well as distinguish certain scenarios, like in this case, vaccination strategies with spike proteins and probably with or without infection.
Thank you so much, Kamala, for your time today and your important research. Before we go, I'd also like to thank the audience for joining us today and their interesting questions, and also our sponsor, Luminex, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until September 9th of 2023, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, thank you so much again, Kamala. Goodbye.
Thank you, everyone.